Good day everyone, my name is Micaela Arsiquito together with Miss Audrey Santos, and we will be reporting our lab activity number 1, which is entitled Microscopy, its principles, parts, use, and care. So these are the group members of Group 6. Miss Melody Calimbol as lab manager, Miss Tabia and Mr. Tabiso as recorder, Miss Santos and Miss Aquito as reporter, Miss Socahel as strategy analyst, Miss San Diego as quality control, and lastly, Mr. Valenton as lab engineer. Also, we have here three objectives, which are the number one, recognize the different kinds of microscope and their functions. Number two is to develop the proper skills of operating the light compound of microscope. And lastly is to observe the structural details of microbial cells. So for pre-lab discussions, Miss Audrey will be taking over.
For the pre-lab discussion, let us familiarize the parts of the microscope. The first one is the objective lenses that magnifies the specimen. Next is the stage or a platform where the specimen is placed for the observation of the different specimens. Next is the fine focus knobs that adjust the focus of the image by moving the lenses up and down. The illuminator or the light source that light through the specimen. Next is the nose piece that holds the objective and allowing for magnification changes. The base that provides stability to the microscope. The power switch that turns the microscope light source on and off. The slide holder where the glass slide is placed with specimens for observation.
Thank you, Ms. Santos. For the guide questions, we have here first, which part of the microscope are used to magnify the objects or specimen? Our answer is the objective lenses because as what we have discussed earlier, objective lenses are used to magnify objects and it has a variety of lenses of observation, low power, high power, and oil immersion lenses. Those lenses helps us to view the specimen in finer details. Second, why is the resolution more important than the magnification? Magnification is about enlarging the picture of our specimen, but resolution is considered more important since it helps us to determine the level of details and clarity of image. And even though the image is greatly magnified, while the resolution is low, we could have difficulties to view the image clearly in our specimen. For the last question, describe the mirror part of our microscope and why would a concave mirror be better than just a plain flat mirror in illuminating our specimen? So our answer is that the illumination part or the mirror part of our microscope is the one supplying for our specimen. And the reason why microscope uses the concave mirrors is that it helps the light to focus on the specimen itself. To compare, the flat mirrors will not focus the light on the specimen, but the concave ones will because of its specified point.
Here are some bacteriology specimens under the microscope. The first one is the Mycobacterium tuberculosis that causes tuberculosis. The Corynebacterium diphtheriae, a throat infection. The Serratia marcescens, a opportunistic or colonic pathogen. The Rhizodium sect of root, a nitrogen-fixing bacterium. Lactobacillus mir, that inhibits the harmful microbes, and lastly is the Shigella dysentery, a dysentery that causing bacteria and a type of severe diarrhea. So for the parasite, the first one is the Giardia lamblia a parasite stage that causing giardiasis or protozoal. The Ascaris female or adult form of roundworm parasite, the hard thick that is dense, the Fasciola hepatica, a liver infecting platform, a Monocystis, a earthworm parasite protozoa, a Hymenalepis diminuta gravid, a tapeworm egg carrier.
So for the botany, we have our six specimen, and as we all know, botany is a scientific study of plants. And first, we have here the Spirogyra vegetative. Um, Usually, they are found in water ponds. And number two, we have here the penicillium. They are found in food spoilage. Third is the third ones is the chrysophytes diatoms mixed with marine forms, and they are a group of algae found in fresh water. Next is the spirulina. They are the ones grows from fresh and salt water. Next is the Leurophyta protococcus. These are known as green algae. And lastly, for the botany, we have here the Helianthus stem. These are found in sunflowers. And for the histology, we also have here six specimens. And as we all know, histology is the study of tissues at a microscopic level. First, we have here the nerve. Second is the mammal duodenum. Third is the ureter. Fourth is a frag stomach. Next is the human prostate. And lastly, the mammal urinary bladder.